Welcome everyone. We're on to the webinar, starting off in CytoSections, validated controls for immunohistochemistry, in situ hybridization, and immunofluorescence staining. The topics we are going to cover today are background. We'll give you what are the challenges, that some of the points that Sean already touched. I'll introduce you to CytoSections, what are the key features and benefits that customers saw when they did the beta testing. We'll go over the performance data, publications, where can you find it on our website, and then we have a short wrap up and a question and answer session. As you all know, one of the critical challenges researchers in the clinical space have is finding appropriate controls, especially validated controls. And you all know there's a very limited supply of these controls. In addition, the new HIPAA rules makes it really difficult and expensive to get these controls. The third is getting inconsistent staining pattern across tissues. And this is nothing to do with the tissue itself, but the way the tissues are treated before you do the staining, during transportation, how it is fixed, what is being used for fixing, how long it has been fixed. All these parameters play into this role of inconsistent staining patterns. And what is the result of that? You have to re-standardize every time you get a new tissue, resulting in wasting time, money, and precious research resources. Another problem that is commonly seen is fading. If you take a tissue section and keep cutting and getting sections of it, there is fading as you get deeper into the sections. And the last point, a challenge which a lot of researchers mentioned, is almost impossible to get controls for mutants and splice variants. Example, you have tissues or you have gene targets which have variant 1, variant 2, variant 3. How are you going to find controls for that specific variant? And to address all these challenges commonly faced by immunohistochemists, antibody developers, we came up with this solution, what is called as CytoSection. CytoSections are FFPE sections of HEC293T cell pellets. These overexpress your target of gene of interest. This overexpressed target is validated by Western blot and immunohistochemistry, and CytoSections work for all different techniques: immunohistochemistry, ISH, and immunofluorescence. What are the key features of CytoSections? First, these are ready-to-use FFPE sections. You get them as unbaked. These are validated, which I already mentioned, by Western blot and immunohistochemistry. Since they are ready to use and validated, you don't have to do any standardization. You can easily be integrated into your lab workflow. And another important point is that these behave like FFPE tissue sections. And this has been tested for manual processing as well as in automated stainers. In addition, they are very convenient. You have DDK tagged. They can easily be detected using anti-DDK antibodies. And there's a special reason why this is so useful. We'll discuss that later. CytoSections are a one-stop solution for controls. You can get positive control as well as negative control. In addition, it also works as a control for all different other applications like IHC, ISH, and immunofluorescence. It's available for all human targets, wild type, mutant, and splice variants. I think the last two are more favorite of our researchers because that is something they have extreme difficulty in finding, especially you want to validated ones. That's a difficult one. It's a different process. And the last is customizable. And we'll talk about it in a couple of slides down the presentation. So now I want to give you a quick idea of how these cytosections are produced. Let's say you send us a request for a cytosections for KRAS. You know that OriGene has one of the largest collection and one of the most oldest collection of cDNA clones, over 80,000 of them. So we take a clone from that. We start off with true OR of gold. What is the typical characteristic of this specific brand of clones? They are sequence verified and they are expression validated. And in addition to that, they also have a MYC DDK tag at the C terminal. One question that often customers ask is, yes, what if I need this at the N terminal and not at the C terminal? Yes, we can do that. That will be part of our customization. So we take these clones. For example, in your case, we want to do for KRAS. We take a clone with KRAS insert, transfect it, culture it for 72 hours, harvest the cells after 72 hours. Then a small part of that cell is pelleted and lysed, and using Western blot analysis, we analyze to make sure that gene is expressed and it is of the right size. Then other part of the cell harvested is pelleted. We embed it in paraffin, cut it into sections, and then again this is verified by immunohistochemistry to make sure that the process works on that specific tissue. So in this case, I just want to mention for immunohistochemistry and Western blot, we use this special antibody that we have generated. It's called anti-DDK rabbit monoclonal antibody. What is so special about it is that whether it recognizes both the DDK, whether it's in the N terminal or C terminal. So one of the things which I keep harboring over and over again is cytosections are validated, and that is why customers and researchers value the most. And I just want to go over again how it is validated. First, as I mentioned, is sequence verified. Number two, expression is validated by the Western blot, that is expression validated. And not only that, it's also validated using by IHC. With that being said, let's move on to applications or performance data. Cytosections are easy and very specific. In this slide, I want to show you that how a cytosection that has been stained for CTLA4 using the antibody Ultramab, as mentioned here, UM8001. This antibody is developed and marketed and manufactured by OriGene. So the first slide, or the first figure, shows you a cytosection that's specifically stained for CTLA4, and this brown color indicates a positive stain. The same cytosection has been stained for CTLA4, but this time we have used the tag antibody. And again, it also shows a very clear positive stain. And the last figure is a HEC293T mock transfected cells, which has vector alone but does not have the insert. And this is also stained by the same antibody what you see here, and you see no staining. The goal of this slide, what it demonstrates, is that cytosections can be easily used, and also it's very specific for staining. The next is regarding staining patterns. Anytime we have an artificial thing that we create, that's always a question, how are the staining patterns? Is it very similar to our tissue, or how is it? So we have done some limited analysis on that, and I'm going to share the data regarding that. So in this slide shows you the staining of BRG1. It is a nuclear protein. So you should say a staining both in the middle of the cell as well as in the cytoplasm. I'm sorry that this specific slide doesn't show the nuclear staining very specifically, but you see a similar stain both in the cytosection as well as the kidney tissue that is stained with BRG1 on both sides. So here, the similar nuclear localization is seen both in cytosections and tissues. Another example is of DCC. DCC is a transmembrane cytoplasmic protein. And again, it is this stained for a DCC with an antibody, and I to clearly see that it's a membrane localization is very clear in these cells. As you see, at the edge is stained, but not the cytoplasm. Similar stain you can also see in the tissue, in the colorectal carcinoma tissue stained for BRG1. In short, it says a similar transmembrane localization is seen in both cytosection and the tissue. The next question is for gene expression pattern. One of the common misunderstandings is that if you have a positive control, every cell in that section will be stained, which is not the case. As you know, we take a plasmid and transfect it into a cell line. Not every cell is going to take it. A transfection efficiency can be maybe 10%, maybe sometimes 5%. And also the gene expressed also depends on the toxicity of that specific gene. So that is what I want to show here is, as sometimes when you see that increasing the cell toxicity, we see that the number of positive cells will be decreased. So when you get to cytosection and when you stain it, surely don't expect 100% of the cells are stained. And that is what I want to show here, the gene expression pattern. There are different kinds of gene expression pattern. For example, if you look at this AFAP, this is a tyrosine kinase activated protein, which you see quite good and strong staining pattern. Whereas when you look at this GPR101, which is a G protein coupled receptor, whose function is not clearly known. Actually, in this case, function is not even known, but we know it's a G protein. Again, you see the staining is very membrane protein. So what I want to show in this slide is gene expression is varied across all different genes. So in short, conclusion is when you look at the staining patterns that localization to some extent we have tested, we can see it. And of course, we also see varied gene expression pattern in cytosections. Now let's move on to the applications. How are these cytosections used by scientists? So one of the applications we've already seen is, of course, our IHC, immunohistochemistry. We have shared a lot of data with you. Here, I'm glad to share you a data that cytosection works as excellent controls for immunofluorescent staining. This is a cytosection that's stained for MAGE-A3C. And here you see DAPI staining. We also see anti-DDK Alexa-4, which is green. And the third one is DDK, anti-DDK with DAPI and Alexa-4, double staining. So this is kind of an example just to demonstrate that cytosections can be used for immunofluorescent staining controls. And the last, I think this is the most wonderful data we have, is that cytosections works for ISH2 or in situ hybridization. Here, our cytosection is probed for ENPP1. And you see these nice red dots here. Actually, these are positive staining as compared to a control probe, which has no staining at all. I want to thank our partners, HistoVis, for sharing this data with us. As I mentioned before about automated stainers, the HistoVis tested these cytosections to work with in automated stainers also. So thank you, HistoVis, for that. So in short, what we have shown so far is that we have shown that cytosections can be used as a controls for immunohistochemistry, ISH, and immunofluorescence, as far as the techniques are concerned. So what are the different applications that you can use for? So let's say you're developing a new antibody and you're testing for a new target and you get a negative result. What does that mean? Well, either you don't have an antibody, that is one possibility, or second, your process is not right. Your process is not optimal. So cytosections are ideal for these kind of situations. As you see here, you can, in the same process, you can use an anti-DDK to make sure that your process is correct. And then if you also see a staining with your for your other antibody, then you know your gene is expressed, but you don't have the antibody. And if you get a positive, then you know that you have an antibody which is very specific for the protein you're looking for. Cytosections can also be used for process control. For example, if you're doing everyday staining and you don't want to use a tissue, then you can certainly use one of the CytoSections. New reagent testing, if you have new reagents in your lab and you want to test for it, again, CytoSections are ideal controls. Or if you're developing a new protocol. In short, what it does, CytoSections, is helps you not to use up your tissue, which are precious. Instead, this gives you a chance to use something else, validate it, and then use your precious tissue resources. One point which I did not touch so far is that CytoSections are customizable. Here I said that CytoSections we can do for any target that can be expressed in HEC293 cells. An example I've given is a mutant protein, which is, again, for example, KRAS mutant G12D. We can express that in this. And in fact, we can verify it and make sure that it's expressed. So if you're screening for an antibody, which you wanted to recognize only the mutant and not the wild type, then we can confirm the fact that we have a mutant in the cell, then you can screen for that specific target. Next is whether can you do it in other cell lines? Yes, we have tried it in CHO cells. We haven't tried it in others, so we can certainly we do that for you. Mixture of cells, different targets. Sometimes there are customers who say, I want multiple KRAS and maybe I need GRP1 or some other protein. So we can certainly mix the cells for you. We can give you variable cell densities. Sometimes we can do 50,000 cells in the core or 500,000 cells, depending on what we can change that. Multiple sections on the same slide. We can put 2 or 3 sections on the same slide. I think we have tried up to 2 or maybe 3. Or last is custom tagging. As I showed you, as a standard protocol, we have the MIG tag at the C terminal, but we can do N terminal, or if you want to do some other tags, then we can certainly do that. The advantage of using what we have done is the MIG DDK tag is that we have antibodies for that. We can provide you that too. So how are this packaged? Generally, you get all the CytoSections. You get it in a box like what you see here. And we get a pack of 25 slides. And sometimes we have requests for blocks. They want the entire block. So we can provide you as a block, but that is a custom product, custom project. We talked about negative and positive controls. And the negative control is basically just the vector alone with no insert and no tag either. And that's a negative control. And the TC4, the 003, this is a positive control with a tagged vector, but no insert. And these are available in packs of 5 slides in each pack. And I said, this is an example of the box you see. So let's move on to how you can find the CytoSections. There are many different ways you can find the targets on our website. But this is one way, is come to OriGene.com and type cytosection KRAS on the top. Once you hit this enter the search button, you'll see these 2 results. And once you see these 2 results, and in these results, there are 2 of them for KRAS cytosection. What you see here is that basically these are 2 different NM numbers. As I mentioned, we can give you cytosections for any accessions or any gene. So again, you can see the clear difference. We can based on the NM numbers. 4 micron sections, 25 slides, and also you can see that it's a tag is mentioned here. We also mentioned what host. So these are again all the things that you can change. And once you move to the next step, just click on the cytosection, this button, and it will take you through the process of online purchase. Have these been published? Yes. Recently, we published it at AACR. There was a poster presented on renewable IHC control for tool cytosections. That was published at AACR in April of this year. You can get a copy of this poster in this link. I can send you this when I'm replying to you in your email when I follow up for this webinar. Same way, we also had a poster accepted at NSH 2022, which just over yesterday. And Dr. Rachel Gonzalez, which I mentioned before, she was invited to present a workshop at NSH 2022. The goal was to how pathologists can improve their control using cDNA-created CytoSections. And this is one of the testimonials that one of our beta testers gave us about CytoSections. Cytosection approach has been very effective and will become an integral part of our workflow. Thanks to all at OriGene for their support, and especially Rachel. As I said, Rachel has been a key player in developing this product and making sure that it works, and who has brought it gave it some light here. And it's very clearly a major pharmaceutical customer who gave us this testimonial in January of 6. As you see, what I want to show is customers like it. It doesn't matter if it's an academic or a pharmaceutical, everybody loves this product. So as a wrap up, I want to show is that CytoSections are ready to use FFPE sections. They are validated. They can be easily integrated in your lab workflow. They're convenient in the sense they are DDK tagged. You can detect them using anti-DDK antibodies. So if you don't have target-specific antibodies, we still can use CytoSections. It's a one-stop solution for all your controls. Whether you want a positive control, a negative control, you want gene-specific control, we can provide you that. It works across a lot of different techniques. IHC, ISH, immunofluorescence is available for all human targets, especially wild-type mutants and splice variants. I forgot to mention that here. I think that is what customers love the most, is the mutant and the splice variants, because they cannot get a validated control for these, especially the mutant and splice variants. And last but not least, it is customizable. You talk to our customer support, our tech support team, and they can tell you whether it is possible or not. But certainly, CytoSections are easily customizable. With that, I want to acknowledge some of the members who actually did this work. Dr. Wei Fu, VP of R&D, and the entire team that actually helped us standardizing this product. And of course, Sean, who introduced me today, and also Saurav for helping us with this presentation. And Histovis, our collaborators, Patrick and Kelsey. If you want to talk to us or contact us, of course, you can write to me. This is my email address. If you're any customer in the globe, you can write here to these email addresses. Here, the contact information for globe other than EU, for EU and for China. As you know, OriGene products are distributed by over 61 countries with an extensive network of distributors. So if you want CytoSections, and if you're outside this country, just find a distributor at origene.com.